Hello, everyone. I'm Christy Risk, Senior Editor at Genomeweb, and I'll be your moderator for today. The title of today's webinar is LongRead Sequencing of Human Transcription Provides Insights into Metastatic Cancer, and our sponsors are LoopGenomics and Twist Bioscience. Our panelists today are Dr. Jianhua Luo, Professor of Pathology and Director of the High Throughput Genome Center at the University of Pittsburgh, and Dr. Indira Wu, Director of Molecular Biology at LoopGenomics. You may type in a question at any time during the webinar. You can do this to the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Luo. Please go ahead.
Okay, thank you, Christy, for the introduction. And today I'm going to talk about the application of the synthetic long-read sequencing developed by LoopGenomics to apply its sequencing analysis to the metastatic colon cancers. This is actually the fact of the human cancers. Actually, according to 2019 CA clinical journals, and that the colon cancer is still one of the most frequent cancer happen in human beings. In 2019, there was approximately 600,880 cancer deaths in the United States, and the top 4 category of cancers incidence is breast cancer, lung cancers, colorectal cancers, and prostate cancers. And worldwide, actually, there is approximately 1 in 6 deaths are due to cancers, and colorectal cancer actually accounts for over 600,000 deaths worldwide. So this actually is one of the most lethal cancers in the cancer category. If we go to the next slide, actually, we actually started the study with 3 patients of colon cancers. And patient 1, we found actually benign colon tissues adjacent to cancers. And then we also have a matched colon cancer, as well as the matched metastatic lymph node colon cancers. In patient number 2, actually, unfortunately, we cannot find the benign colon tissues adjacent to cancer, but we have the cancer tissues along with the matched metastatic lymph nodes. And patient 3, actually, we actually have all 3 types of tissues. And after collecting these 3 tissues, we perform the so-called loop sequencing through the collaboration with LoopGenomics, and first through the barcodings, and then enrichment of 4,000 genes that were shown to be frequently involved in chromosome rearrangements, deletions, and mutations. And subsequently, we do a loop-seq library preps and then sequence Illumina sequencing and then the de novo assemblies, and perform a so-called quantity isoform mapping. And this is actually a basic scheme of how to perform this loop-seq, and actually Indira would give you a more detailed analysis and a description of the process. Basically, is a mRNA being captured by oligo-DTs, and then bounded with the so-called barcode adapter enzyme complex, and subsequently to it being enriched by a process of 4,000 genes throughout all the exons of those genes. And after that, we perform a so-called amplification and cis-strand barcodings, and subsequently a fragmentation will basically able to fragment those barcoded strands. This barcoding process actually only happens in cis-strand, but not in the trans. That actually allows us to identify which cDNA is actually belong to which molecules. As a result, we are able to do a de novo assembly. Actually, we recover the entire molecule length. So the first goal of this analysis is to identify the primary and metastatic tumor novel fusion isoforms. And previously, actually, we had enormous difficulty to identify the isoforms for the fusion genes because the rarity of these fusion genes, and also it's difficult to piece them together through the short read sequencing. So to perform a so-called screening of the novel fusion gene isoform, we basically apply four criteria for the filtering process. One is so-called if a cis direction, gene A and gene B, basically in the same orientations, and gene A is upstream of the gene B, then has to be separated by more than 40 kilobase on the left upper scenario. And if there is a trans orientation, such as gene A and gene B in so-called trans direction 1, the gene A and the gene B actually has a different orientation, and then we just simply, any distance is fine. And the trans orientation on second scenario is the switch of the position of gene A and gene B in the chromosome. The gene B actually is upstream of the gene A. However, in the real RNA transcripts, end up with gene A is in front of gene B. In this also called a so-called translocations. And the scenario 3 of the translocation is the different kind of different chromosome set actually transfer from one chromosome to another one, like chromosome 1 transfer to chromosome 15, produce a chimera type of RNA. So in this, whenever those fusion fit within these four criteria, any one of these stuff, and then we call a likely a fusion resulted from the chromosome arrangements. So based on this criteria, after the quantity analysis, we basically found a number of those fusion transcripts present in the tumor samples, which is 1T, 2T, and 3Ts, but not present in the normal colon samples, the match one. And in this case, we found that actually the red one indicates that the fusion gene has been validated by Techman RT-PCR and Sanger sequencing. And those black indicate the fusion gene yet to be validated. Some of them actually is false, likely, but not entirely sure. The other one actually never been tried to validate them. So in this case, we found like SATM-BPL1 and FAST1 actually across four samples of tumors and metastatic samples of colon cancers, and another one is in the 3T and 3LN. LN means actually lymph node metastasis. In this case, you will find that the VAP-BP GNAS was actually fused together, and both in the primary cancer samples and the lymph node samples. So I show you one of the example of the fusion occurring in colon cancers, which is so-called receptor tyrosine protein phosphatase kappa, and another one actually is as a monocyl CoA decarboxylase 1, and the PTPRK actually is a receptor tyrosine phosphatase, actually plays a role in the phosphorylate the tyrosine residue in those growth factor receptors, therefore plays somewhat a tumor suppressor gene role. And the ECHDC1, which is so-called as a monocyl CoA decarboxylase 1, on the other hand, is like a gene involved in fatty acid metabolisms. And you can see them, these two genes actually separate by 624 kilobase, and they are same orientations. In the fusion, what we found is that the PTPRK lost about 8 exons from the transcriptions, and then hook up with the last 3 exons of ECHDC1 and produce some kind of chimera proteins, and which actually is more like a truncated type of PTPRK. In normal situation, the PTPRK, as you see from the right-hand side, is exon 1 to 13 produce a so-called receptor tyrosine phosphatase. There is a large chunk extracellular domain, which is 752 amino acids, and with one transmembrane domain, which is about 24 amino acids, and as well as a large chunk of cytoplasmic phosphatase domains. And however, after the fusion, it produces a huge truncation, and therefore only about 387 amino acids left in the proteins, and produce a frame shift. As a result, eliminate the open unit frame of the ethylmalonyl-CoA decarboxylase. Actually, that about 41 amino acids actually produced is the frame shift, and therefore this resulting a truncated protein basically produce a so-called functional knockout of both genes, PTPRK as well as the ECH-DC1. So subsequently, we perform a validation analysis using the Tegman PCR, RT-PCR analysis. We design a probe right at the juncture of the fusion transcript, and then a juncture on the with a primer right next to the probe, and as well as the primer for the EC-DC1, and then produce a small RT-PCR fragments. In this case, we found that this gene again basically confirmed that the loop-seq accuracy. In this case, patient 1 basically all are negative in terms of this transcript, and patient 3 in this case only the metastatic lymph node cancer contain these fusions, as you can see from here on the right-hand side on the validation sets. So the metastatic RT-qPCR, you can see from there. And then this is a second example of the fusion gene we detected. Actually, in this case, we found 2 samples are positive for these fusion genes. One is actually involving a so-called guanine nucleotide binding protein subunit alpha, and then the other one actually is a WAMP associated protein BNC, which actually is a vesicle trafficking proteins. And basically, this is a VAPB interact with WAM1 and WAM2, and basically is essential for the vesicle trafficking inside the cells. So this actually is gene is separated by approximately 388 kilobase, and in the same orientation in the fusion, you can see that the VAPB actually lost all the exon except exon 1, and then hook up with exon 2 all the way to exon 12 of the GNAS. And the GNAS actually is a protein is more like most of the functional domain is in the C terminus, and is a G protein alpha. Basically G protein, nucleotide binding protein, right there, and then the N terminus is more like a vector domains. Many actual protein interact with this particular N terminus. For the VAPB1, actually, it contains a so-called major sperm protein motif and coil domain and transmembrane domain actually. But after all of these fusions, all those domains gone except a partial retain of this MSP, the so-called major sperm protein motif right there, but very much intact G alpha subunit domains from the GNAS. So the function of this one is not clear. However, it looks like the vector all wipe out. As a result, the function may extensively being changed. So we do the same validation on this particular protein, as you can see from here. Actually, interestingly, in the loop-seq, we didn't find any of these fusion transcript in patient number 1. But however, when we do a RT-PCR analysis, we found actually it's positive in these fusions, for this fusion in these 2 samples, the 1T and 1LN. On the other hand, we also confirm that patient number 3, which is, we know that they are positive for this fusion gene, and also turn out to be positive as a result. So this suggested that the loop-seq sequencing, even by enrichment, may be still not sensitive enough to detect all the fusion genes. Many fusion genes may be still probably left undetected as a result. So on the other hand, if we increase the depth of the sequencing, more fusion genes may be uncovered as a result. So intermediate summary of this one is a pro-capture coupled with long-read sequencing. We could identify the novel cancer fusion isoform in both primary and metastatic cancer samples that are not present in the normal colon tissues. The second part of this seminar, I will like to discuss the isoform quantifications to distinguish cancers versus the normal tissue as well as the metastatic cancer samples in the lymph nodes. So first thing we do actually is try to perform a DE gene analysis, basically perform the, as you see on the left panel right there, the differential expression between the N means the normal colon tissues, T means the primary cancer samples, and LM means the metastatic sample in the lymph nodes. And based on either they are more expressed in the lymph node or more expressed in the normal or more expressed in the primary cancer sample, we have like 8 different kind of category from here. And we found that there are approximately 2,682 genes actually being differentially expressed between these samples. And then we group them together, as you see in the heat map on the right-hand side. Then the second question that we ask is, can we utilize this panel of differential expressed genes to segregate the samples of colon cancers, normal colon, as well as the metastatic cancer sample in the lymph nodes? As you see by a hierarchical clustering on the left-hand side, you can see that these differential expression genes actually could able to segregate the normal samples, 1N and 3N here on the left-hand side, from the rest of the cancer sample, either the lymph nodes or the primary cancer sample, 2T, 3T, and 1T. But the segregation between the primary cancer samples and the lymph node metastatic samples seems not as good as I would like to think. And subsequently, to corroborate this analysis, we perform a so-called principal component analysis. We found that actually these differential expression gene could segregate the normal sample, the green dots on the left upper column corner, and then compare with the one that on the right-hand side right lower corners, and you can see the primary cancer sample as well as the metastatic lymph node sample. However, they actually didn't separate very well between the T samples and the LN samples. And then we perform a Pearson correlation analysis. As you can see that the 1N and 3N, the normal sample, somewhat will segregate them together and actually produce similar kind of correlation, Pearson correlation with each other. But however, if you look at the T sample and the LN samples, they somewhat like has very similar color, and it indicates that the segregation of primary cancers and the metastatic cancer in the lymph node is not ideal. So based on this, we concluded that the gene expression alone is competent in segregating normal colon from cancers, but somehow less efficient in terms of segregating metastatic cancers from the primary cancer samples. And then we look at the isoform expressions, differential expression between the lymph nodes, the primary cancer, and the normal cancers, normal tissues. And we also we found 7,000 events, so actually looks more robust. And also we found to segregate them into 1N, 2N, 1T, 2T, and 3T, and make a heat map on the right-hand side. When we perform a hierarchical analysis to identify whether this differential expressed isoform can segregate the benign sample versus the colon cancer versus the metastatic colon cancer, we found that actually performed the job very robustly. And you can see the 3N, the benign samples, 3N and 1N all clustered together. And the metastatic samples, LN, 1LN, and 3LN all clustered together. and the primary cancer sample, they're all together. And when we perform the principal component analysis, and we found that these sample actually segregate extremely robustly. You can look at the primary cancer samples all on the left upper corners, and the metastatic lymph nodes all on the bottoms of the box. And then if you look at the normal sample, is very far away from the tumor samples. And then we will look at the analysis of so-called the Pearson correlation analysis, basically look for how they correlate each other by the coefficient efficiency. And then we look at it, actually, we found that the normal sample actually closely unrelated or actually inverse related to the tumor samples, and the lymph nodes and the primary cancer sample, they all have similar colors, and those lymph node metastatic samples also have similar colors. So actually produce a robust segregations. So we concluded that the differential expression of isoform produce a robust segregation of normal tissues, colon cancers, and metastatic colon cancers. And the segregation between primary colon cancer and metastatic colon cancers was only feasible with isoform expression data, but not based on the gene expression alone. So we know that the DE gene event has a 2,682, and then the DE isoform event actually has more than 7,000. So we overlap these two group of things, isoform differential expression versus the gene expression, and produce a so-called Venn diagram here on the left lower part of the slides. And you see the group 1 actually, that means they have a gene expression differential expressed in 3 groups. However, they had no change in isoform switch. Basically, no switch isoforms. And group 2 is the gene expression differentially between the 3 samples, also have a differential expression isoforms. Group 3 is opposite. Basically, there's no change of expression in the gene overall. But however, there's so-called a switch isoform within the genes. So we want to identify what's the role of these groups in playing in segregating the cancer samples. In the next slide. So first thing, we want to look at the group 1, which has a gene differential expression but no change of isoform distributions. And the segregation actually very bad. In this case, the lymph node and somewhat still okay, but the normal samples and the tumor primary cancer sample actually intermingle together. And you can see from also in the principal component analysis, and you can see that the PC1, PC2, and then you can see that the green dot actually intermingle with the metastatic lymph nodes, and as well as the red dots, the primary T samples. And if you look at the Pearson correlation analysis, we found also similar things. Like 1N sample actually found a lot of correlation with those T samples, LN samples, etc. So somehow the ability based on the gene expression without the change of isoform distributions causing a insufficient to distinguish normal colon cancer as well as metastatic cancer samples. And then second, we go ahead to analyze group 2, which is the gene that has a differential expression among the 3 groups, but also has the distribution changes of isoforms between the 2 samples. What we found actually is somewhat improvement in the hierarchical clustering. As you can see from here, that 1N actually unrelated to any samples, and then however 3N somewhat related a 3T, and the 3 lymph node meta sample are clustered together. And when we perform a principal component analysis, we found that the lymph node metastatic also similarly cluster with the primary cancer sample of colon, but that are segregated from the normal colon tissues. So suggesting that this segregation between the normal and the tumor tissue probably efficient, but may not be very efficient in distinguish the metastatic and primary colon cancers as a result. So if we look at the Pearson correlation analysis, similar thing we found it, and then between the between the 1N and 3N versus the other primary cancer samples and metastatic lymph node samples, they are not related. They really well segregated. Coefficient completely, inverse, something like that. But if you look at the T sample versus the LN samples, they are very mixed up. So basically, they are not really a good segregation between the metastatic samples and the primary cancer samples. And then the next thing I want to see is, does the group 3, which is the group of gene with isoform changes, but no overall gene expression alterations across the board, which is actually accounted for the most largest group of the genes, which is approximately 4,000 genes there, and you can see that actually, in fact, it's 4,000 isoforms, sorry. And then in this case, we found that actually, based on the hierarchical clustering, we found that the so-called isoform segregation able to produce a so-called complete segregation between a benign, metastatic, and primary cancer samples in the hierarchical clusterings. And then we confirmed that in the principal component analysis, you can see from there, the normal sample, the green dot, actually is all the way to the right-hand side edges of the box. And the metastatic samples is on lower corners of the left-hand side. And the primary cancer sample is the upper corner of the left-hand side. So as a result, it has a very good segregation between the samples. And if you look at the Pearson correlations, and you can see these actually similar changes, you can see that the N sample actually unrelated with the so-called T and LN samples. And however, the T sample, they all basically have similar colors, and the LN sample, they all have similar colors. As a result, they are basically well segregated based on the analysis. Because of these isoform changes actually is undetected by gene expression analysis, we call it silent isoform switches. Actually, it alone could produce a robust segregation of normal colon cancers and metastatic colon cancer samples. So this is the next slide. Is show you some of the examples of these so-called silent isoform switchings. One of them, the first one on the left-hand side, is called so-called prostaglandin E synthase 3, which actually involved in prostaglandin metabolism, is involved a variety of those inflammation reactions. And you can see that this gene actually is not differentially expressed between the normal and the tumor samples. However, this particular isoform produces a large difference between the samples of cancers as well as tumor and normal samples. And you can see the normal sample is somewhat low expressed, and then the tumor sample has a dramatically high expression level. The second example is RUFI1, which is a run domain and ring finger protein, which involved in so-called endoplasmic reticulum trafficking. And this protein actually also is not expressed, it's evenly expressed in both normal and cancer samples. And however, you can see that its expression is one of the isoforms is extraordinarily being shoot up in terms of the expressions. And the next example, TRMT6B, which is so-called tRNA methyltransferase, which is involved in the stability of tRNA. And you see the expression level of this gene is not changed, but the isoform has big changes over here. And the last one is so-called a YAP1, which is a effector of hippo signaling pathways and being a target of the drugs. So actually, this isoform in the cancer actually being shoot up dramatically in comparison with the other isoforms. And gene expression-wise, they basically same between the sample, cancer samples and the normal tissues. So when we go in the individual isoform of a gene, and I think CD44 is a good example. CD44 is one of the most frequent metastatic markers for human cancer, including like colon cancers and prostate cancers and even liver cancers. And these are all expressions found. However, in this case, we found the expression of CD44 actually not changes, but the isoform change. You can see that this, if you look at the right-hand side of this particular gene, is amazing that one gene, it produces 26 isoforms, which actually means that these 26 different proteins from the same gene. They're all different. I mean, think about the diversity produced by one gene, and why they produce so many, I don't know. So my guess is that some of them actually adjust their survival based on this. So if we look into this particular example, 1N, 3N is a normal samples, 1, 2, 3T actually is the primary cancer samples, 1, 2, 3 LN is the lymph node metastatic sample. You see that the green arrow here show that that's the gene isoform that appear only present in the tumor sample, but not the normal samples. The grayish, you can see from that one, 5 out of 6 actually have this isoform, but not present in the normal. Another one actually is more the orange, also indicated by the green arrows, show up in the cancer samples, but not in the normal colon samples. In fact, a couple of more, but I do not want to delve on these examples. Another example actually is somewhat changes like this ATP sodium potassium pump. And itself actually has about 5 different isoforms. Normally, only 2 isoforms express produce a protein, which is, you can see from the right-hand side, which is the NM0016023.4.1, and the other one is XM017001361.1, and that's normally expressed. However, in the cancer samples, you will see appear a new isoform being there indicated by the green arrow, which is produces somewhat like subtle changes of the protein structure because of additional 32 amino acids being present in these isoforms. And then when in metastatic, another isoform show up, actually produce yellowish color, that isoform. Actually, these two actually, even though the greenish and the yellowish isoform looks like the same amino acid sequence, but actually they are not the same. Actually, the isoform, there are 3 amino acids different in the N terminus, so they are not the same protein after all. So another example I want to show you is that the beta-catenin. Actually, beta-catenin is one of the tumor oncogenes actually involved in a variety of human cancers, particularly in the hepatocellular carcinomas. And this gene normally expressed, actually, you can see that only 5 isoforms being present in normal tissues, 5, maybe sometimes 4 in this case. But however, once become malignant cells, actually the cancer cell produce variety of these isoforms. It actually produces 5 additional more isoforms. You can see from those green arrow, either up or down. In fact, if you look at the overall expression of these beta-catenins, they are not changes. But however, is underneath the this so-called seemingly uneventful expression level is there's a seismic shift in terms of isoform composition. They produce a different kind of protein there, even though the expression level of this gene is kind of similar. So it suggested that there may be a new mechanism for this particular important oncogene to drive the cancer to growth and to invade. So based on this analysis, we produce a so-called intermediate summary, and with differential isoform, but not the gene expression, is able to differentiate the primary and metastatic cancer samples. The differential expression gene is good to distinguish normal versus cancer overall, but when it distinguishes tissue subtypes, the survival environments, and the isoform seems to do a better job. And second is that isoform diversity switching is greater in tumors in comparison with the normal tissues. In all the examples I show you, actually, you found that when it switches to a cancer type of tissues, there always has more diversity in terms of isoform expression compared with the normal tissues. As a result, that may explain this kind of so-called normal, means that there is a diversity, means that they also change the structure of the protein as well. That because it is probably because the structural change of the protein that drives the cancer to metastasize and to growth. And the third thing, take-home message, is that the gene expression study are blind to silent switching. In fact, silent switching looks like to be the most abundant changes gene expression in comparison with differential gene expressions. And in this case, without the isoform analysis, you miss a huge number of the alterations in human cancer samples. So in the third part of this webinar, I will talk about the SNP. Basically, is so-called mutation. Likely, it could be a result, a isoform-specific expression of certain SNP. One example, actually, we actually analyzed the samples of these tumors versus the normal, and then we found actually in overall we have more than 1,000 SNPs across all different samples, and that are likely to be, well, I would call it mutation likely because they're not present in the normal. And interestingly, we found that the question is whether this mutation is expressed in specific isoforms. And then if you look at this one, the TP53 and G245D mutations, we found that actually it's present in the long reads of this NM001276676111, and in actually is, however, these two isoforms is somewhat similar. There are some, what difference between these two actually is lies in the 5' UTR regions, not in the proteins. And then we found that actually one of the more frequent mutation in colon cancer is BRAF and V600E. You can see here, the BRAF actually has only 5 isoforms, and is lucky. And interestingly, we found that the distribution of these V600E mutations is not evenly distributed across all the isoforms. In fact, it's always concentrated on certain isoforms. In this case, is NM004133.5. Each of these isoforms, interestingly, produce different proteins. They are not the same protein, even though they are the same genes. If you target at this protein by the drugs, you need to know what protein you're targeting at. Actually, you have to basically target at the right structures of the proteins, and each structure, the structure of each protein actually are different. In this case, so it may be food for thought is that when we analyze the mutation by exome seq, we may miss a lot of information, what protein we are targeting at. And then if you look at this 3LN, which is metastatic lymph nodes, and patient number 3, and then same thing, or the same place, actually, they always concentrate in certain isoforms, but not all other isoforms. So this case indicates that when we design the drugs, we need to design the drug on a specific protein, not just a gene alone. Otherwise, it could likely hop on a wrong boat. So does the mutation also switch isoform? Yes. This is another sample of colon cancer. In this case, you can see the KRAS mutations, basically G12V, happen in colon cancer patient number 2, primary cancer sample 2T, and then by the cancer sample 2LN. So when we analyze the primary cancer samples, we found that only one mutation being present in a NM 0336 0.3. And however, when we analyze this mutation in the metastatic sample, it switch. Somewhat like the NM 03336.0 has somewhat like, in fact, they actually have more mutation being found, indicates that being enriched in some way compared with the primary cancer samples. And also they switch the isoform from a 33360.3 to 004985.4. Basically, these actually are different proteins. It's not the same protein after all, because the 3360.3 actually is 189 amino acids, and the 4985.4 actually is 188 amino acids. The difference between these two proteins actually about 20 amino acids, 15, 20 amino acids, whatever it is. And then they actually have produced a structural difference, and then may actually evolve in somewhat subtle difference in terms of signal transduction. So the conclusion of this session is that the SNP are found in specific isoform, and usually a dominant certain isoform, but not evenly distributed to all others. And the change of mutation isoform may result in structural changes of the mutant proteins. That may be it has implication that how do we design a small molecule drug to which isoform? When we perform an assay, which isoform, which protein you want to assay on? That's a very important part. So in summary, the webinar showed that the probe capture coupled with synthetic long-read sequencing with high accuracy will provide an important tool and a new level of granularity to analyze gene expression regulation. And next step, we're going to perform the cell-specific isoform characterization to understand isoform and mutational distribution in a heterogeneous cancer cell populations, because in order to understand the driver, we need to make sure understand what is the makeup of the cell population in the cancer samples. So in this study is supported by the grants from National Cancer Institute and Department of Defense. And we are very fortunate to have the collaboration from Twist Bioscience Incorporation and LoopGenomics, and they provided the basic foundation of the technology to allow this type of analysis to happen. And next, I think I will transfer to Indira for her talk.
Thank you, Dr. Luo. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Wu. Please go ahead.
Thank you. Thank you, Christy, and thank you, Dr. Luo, for the wonderful presentations that you just did. So I want to say a few words about the technology innovations that went into the work that you just heard. So the problems that we set out to solve is that with short-read sequencing, which is a very popular platform and ubiquitous, is that the DNA and RNA molecules that you want to sequence is often much longer than the read length of the sequencer. And so one of the first things that you do in sample prep is to cut up the long pieces of DNA and RNA molecules that you have in your samples. Although by doing so, by cutting the molecules into a size that's appropriate for the sequencing platform during sample prep, you lose that long-range information that would otherwise provide important insights into your samples. And so in the case of Dr. Luo's work, that we just heard, you lose the ability to confidently call isoforms and identify any kind of alternative splicing events. And in addition, to get this kind of long-range information on a short-read sequencer, you would need to over-sequence to obtain overlapping coverage on your target DNA or RNA, which just remains too cost-prohibitive on any platform. And so what we really need is a way to focus the sequencing dollars on specific genes or regions of interest, and really sort of zoom in on the data collected. And so the solution that we're proposing is sort of a combination of sample prep kits. And so what we offer at LoopGenomics is a synthetic long-read sequencing solution using your existing Illumina short-read sequencer. And so with our sample prep method and software analysis, you can transform your short-read sequencer into a long-read sequencer by simply just using a different sample prep kit. In this study, we also coupled with a highly efficient probe capture method from Twist Bioscience to really drive that sequencing cost down and focus the analysis only on the genes of interest. So a few more words about the synthetic long-read sequencing technology that we call LoopSeq that we developed at LoopGenomics. And so the sample prep takes place inside of an Eppendorf tube without the need of any additional hardware. Once the target nucleic acid molecule is tagged with a unique barcode, in this particular study, it would be the first-strand cDNA that's transcribed from RNA. So each of these DNA molecule is amplified thousands of times using PCR, and then each molecular copy with its barcode would have this barcode sequence randomly copy and pasted within the length of the molecule. And so once that barcode insertion takes place, the long molecules are then fragmented and prepped just like any other short-read sequencing library. And on the Illumina sequencer, the barcode, as well as the adjacent segment of the DNA that the barcode was inserted into, are read. And after sequencing, we have our a software analysis pipeline that would bin the short reads that share the same barcodes for de novo assembly to obtain the full-length sequence of the starting long molecule. And so in this study, we coupled the power of synthetic long-read sequencing from Loop with the cost-saving benefit of target enrichment from Twist Biosciences. So traditional sort of DNA or probe synthesis is using column-based phosphoroamidite chemistry, and it relies on standard laboratory plates to generate these probe molecules. While it's effective, this method is slow and expensive. And since they're done in sort of plates at a time, it's very difficult to provide cost-effective and custom NGS solutions using this type of analysis. And then on the other hand, what Twist synthesizes probes using the same phosphoroamidite synthesis process, which has been used successfully in the industry for decades. But what separates Twist is the ability to shrink that chemistry down onto a silicon chip. And so on each of the silicon chip, it contains 9,600 clusters, and each of this cluster has the ability to synthesize 121 unique oligos. And so this is essentially oligosynthesis at massively parallel scale. So think of it as how, in terms of the innovation, this is equivalent of essentially the Illumina inventing the massively parallel sequencing, but on the DNA synthesis. And so by combining this cutting-edge silicon-based manufacturing process with a robust and scalable oligosynthesis chemistry, Twist can provide you both with the customization that you need for your experiment, as well as the high-quality data at an affordable price point. Oligosynthesis on a silicon chip is printed at about a femtomole scale, which is actually insufficient for many applications, including target enrichment. So in order to obtain oligos in sufficient mass to be used in NGS, Twist has developed a proprietary amplification method to amplify the probes in an extremely uniform manner. And so this is extremely important because you can imagine if the amplification bias is introduced due to GC content, as shown on the left, it can translate into non-uniform capture and non-uniform sequencing data. And so to that end, the amplification method that Twist developed maintains that uniformity during amplification. And this results in a highly uniform capture pool that is independent of the GC content of the probes. And a uniform capture results in efficient and accurate sequencing, which is critical for labs looking to maximize their sequencing throughput and reducing their sequencing cost. And because the probes undergo an amplification step, Twist target capture probes are unique in the enrichment market as they consist of double-stranded DNA. And so during the melt step, and then the double-stranded probes unwind and becoming two independent probes of complementary sequence. And this enables the user to capture both the sense and the anti-sense of the adapter ligated molecule. This means you can capture, as opposed to competitor, which at best I think offers a single-stranded DNA probe, which means if you use those probe sets, you will only capture half of the mass. In contrast with the double-stranded Twist probes, you can capture and sequence the most complex and diverse library possible. So putting all this together, the data from today's study was generated by taking the total RNA extracted from tissue, either normal, column, or metastatic cancer, and simultaneously transcribe and barcode the mRNA. We then use the Twist target enrichment kit to pull down mRNA from specific genes of interest that has been shown to have important implications in cancer development to really concentrate on that sequencing power. The captured long molecules are then prepped using LoopGenomics sample prep protocol and assembled using the de novo assembly pipeline. And with that, the panelists would like to take any questions that the audience has.
Great. Thank you, Dr. Wu. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, joining us for the Q&A is Brian Hogland, Product Manager for Next Generation Sequencing at Twist Bioscience. Okay, Jianhua, the first question is for you. Can you tell us a little bit about how, more about how you define isoform switching? For example, does it involve different promoter usage, exon inclusion or exclusion, or other criteria?
Yeah, I mean, so when we define the switching, it's based on the available isoform, which are mapped with quantities, and then to quantify them, and then compare between the samples. So promoter-wise, I believe most of them are the same. Certainly, there are some, based on analysis, we also found some alternative promoter being used and produce a novel type of isoform, which actually we have not discussed here. But those all isoform switching actually shown is a known isoform. In other words, certain exon being used and not being used, and that's compared between them.
Jianhua, did you compare LoopSeq to, for example, Oxford Nanopore Technologies' long-read sequencing? And if you did, can you tell us a bit about the advantages or disadvantages that you saw with the two different techniques? And Indira, if you want to jump in on this, that would be great.
Yeah, we actually also perform Oxford Nanopore sequencing a little bit, but not the same sample, so I cannot tell you that directly. And Oxford Nanopore actually also sequence very long, but the unmapped rate is fairly high, so probably is indeed different type of technology. One advantage of the Oxford nanopore is that the cost-wise a little bit less because of it's basically the sequencing, or just by in a so-called onion type of a small instrument. And in comparing the LoopSeq actually a lot more extensive, and in terms of accuracy, seems that apparently the LoopSeq is from Illumina accuracy, so that's the excellent part of it. For Oxford nanopore, we try to perform mutation analysis. We all fail because we could not tell that too many of those mutations happens.
Yeah, so I do want to sort of, since Dr. Luo brought up the issue with error rates, I do want to say one of the biggest advantage of using LoopSeq synthetic long-read prep is the error rates. So I think Dr. Luo mentioned that. So the first point is that since the libraries are sequenced on a Illumina sequencer, we are piggybacking on the excellent error rate of Illumina sequencing. Although on top of that, because this is a synthetic long-read technology where the long reads are assembled, we have the added advantage that we have consensus sequence correction. So we can actually drop the error rates from the Illumina sequencer down 40-fold lower compared to what you would get with a short read prep. And so that's usually when you find a SNP or deletion event or kind of isoform switching, it is supported by many, many, many reads.
Great, thank you. Indira, can you tell us a little bit more about how the barcodes are distributed across the molecule in LoopSeq, and also how long the barcodes are?
So the barcodes are about 12 bases, and essentially the barcodes are distributed so internally using a combination of enzymatic reactions, that is intramolecular enzymatic reactions.
Great, thanks. Jianhua, do you suspect that some proportion of the isoform differences that you saw could derive from tumor purity issues or a degree of lymphocyte infiltration?
They're always possible, because when we look at the lymph nodes and also look at the primary cancer samples, of course you see the distribution of lymphocytes quite a bit different there. And however, if we look at the tumor versus the lymph node metastatic, the distribution is similar. So actually, I feel good about that part.
And when you detect novel fusions, do you still detect normal transcripts from those same genes? And if so, what's the relative expression of the fusion transcript compared to the normal isoform?
Fusion relatively rare. So I would say that probably I would say one real fusion and then probably 10 normal there. And the reason for that is because not all the cancer cells have the fusion. Only a subpopulation, maybe 10-15%. So in that case, it's not that easy to detect the fusion gene because of the heterogeneity of the cancers. Particularly also mingle with some of those lymphocytes and vascular, endothelial cells, or like that.
Could you detect these fusion events with next generation sequencing?
Yeah, I think so, because we try to validate those fusion events, and interestingly, we found many fusion actually listed on the table in black are not really inside the intron region, means like coming from like RNA recombination, and those are so usually not able to validate. And only those gene fusion genes looks like it's coming from the splicing of two different exons, two exons from two different genes, and then the probability of being validated is much higher.
And have you found that is there an ideal or maximum size of the input DNA or cDNA?
I can take that question. Oh, sorry, go ahead, Dr. Luo.
Yeah, I think the deeper probably is how deep is that's probably is the crucial part. And in fact, we have a couple of fusion genes we know that's present in those samples, but we still cannot capture it. That's certainly, I think that depth is an extraordinary problem for the identifying a novel fusion genes. And actually, by that, it means that we show you that 2 samples actually are negative in loop-seq, but actually when we do a Q segment, Q RT-PCR, they show up as good as the other samples, obviously. Even by enrichment of pole capturing, and still has some still a long way to go to actually able to capture all the fusions in cancer cells. But I believe that they means the fusion genes are very common in cancer cells, in fact, and we found them actually very easily detected. If you once you identify them by Tegman and by FISH, they could all be found.
Indira, did you want to follow up with that?
Yeah, I just want to comment that it basically comes down to, so the question is about how much input, and it basically comes down to what the rarity of the event, like Dr. Luo mentioned, as well as the size of the panel that you're trying to capture. So you can imagine that if you have a very rare event, but a very large panel, because you want to check thousands of genes at the same time, then you would need a much deeper sequencing depth and sequencing many more molecules to achieve that. And the more molecules you sequence, the higher the input you would need.
In fact, actually, when we starting this project, at one point actually we didn't put enough samples. I mean, cDNA sequencing, depth sequencing, and end up we didn't find many fusion genes. And then later we then more samples, I mean, sequence more depth, and then it show up all, means quite frequent. So actually, it's a depth issue, more like. Agree with Indira, 100%.
All right, thank you very much. I'm afraid that's all the time we have for today. We'd like to thank Jianhua Luo, Indira Wu, Brian Hogland, and our sponsors, LoopGenomics and Twist Bioscience. We have many more questions in the queue than we've been able to answer today, so we will follow up with our experts on the unanswered questions. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.